So first thing we're going to talk about is computer science. Specifically, there's something called dynamic programming. All right. So I don't know how familiar everyone is with coding, but this is like a special like strategy for making algorithms in computer science. All right. So the idea here is that you use, the strategy is used for solving optimization problems. So like shortest path, minimum edit distance, etc., and is extensively used in bioinformatics for sequencing analysis. So how does it work? Okay. So basically, first of all, the first way you want to use DP is to divide your problem into smaller problems of the same like form. So for example, if we want to figure out what the say 100th Fibonacci number is, all right, we know if we know the 100th, we know that to calculate the 100th Fibonacci number, all we need to do is add the 99th and the 98th Fibonacci number, right? So in essence, we broke down this big problem of finding the 100th Fibonacci number into two smaller problems, which are finding the 99th, 95, the 99th Fibonacci number and the 98th Fibonacci number. All right. So again, these two problems have the same form of the big problem, and so that's why we can divide it. And then of course we can continually divide the 99th number into the 98th and 97th. And if we keep going all the way down, and you already know what the 0th and the 1st Fibonacci number are, and by doing that, you can calculate what the 100th one is. All right. Again, this is just one way of looking at strategy of looking at how to solve these problems. All right. Yes. So the idea here is that instead of just like trying to brute force it, I guess, all you need to do is you can store the like answers to all the subproblems you've needed to solve. So anytime you run into a subproblem, you're going to try to solve it. If you don't have enough information to solve it, you'll probably go to another subproblem. And once you've already solved a subproblem, you're going to record its solution, put it in somewhere in your computer. And then if you need to solve that subproblem again later, you can just directly pull it out of your computer. All right. Again, so here, let's see if I can draw a more like schematic of this. All right. So instead of using 100 though, to save myself, I'm going to use say Fibonacci, the 4th Fibonacci number. All right. And so how a DP program would do this? Oh my gosh. My drawing with a trackpad is horrendous. What the DP program would say was like, all right, to solve the 4th Fibonacci number, I just need the 3rd and the 2nd one. All right. So, okay. So that means I'll split this problem into two subproblems, which is f of 3 and f of 2. All right, cool. So then it goes, all right, do I know how to solve, say, let's go f of 2 first. Do I know how to solve f of 2? And the answer is no, I don't. I only know, oh, by the way, I should probably record that what we do know already is, okay, what we do know already is that f of 0 is 0 and f of 1 is 1. All right, we don't know anything else yet. All right, so we go, okay, do I know what f of 2 is? No. So I'm going to go down here, f of 0. All right, cool. And I keep going down. It goes, oh, do I know what f of 0 is? Oh, yes, perfect. I do. All right. So now the computer, what it's going to do is going to take away f of 0 and just replace it with 0. All right. Again, how does it know? Well, it goes back to its like dictionary, I guess, and it sees that f of 0 is in fact 0. So it can replace it. It does the same exact thing with f of 1 here. And then after, it's like, okay, cool, I've solved both subproblems. So now all I need to do is add it together to get f of 2. All right, so they got f of 2, perfect. All right, and now it's like, okay, this is the first time I solved f of 2, so I'm just going to add the entry f of 2 equals 1 into my dictionary. All right, and it's going to keep going down like this. All right, so it goes back down to f of 3. All right, so f of 3 is just f of 1 plus f of 2. All right, cool. Yes, so it goes f of 1. Oh, I already know the solution to that problem, so I'm going to cross it out. And f of 2. Oh, I already know the solution to that problem, so I'm going to cross it out. and replace it. Okay, now I can solve f of 3. So f of 3 is 2. All right, so now I can cross it out. Again, since it's the first time I'm solving this solved problem, I'm going to replace a 3 with a 2. And then I'm going to go all the way up to f of 4, which we can now see is 3. All right. Again, you maybe think, okay, this is kind of dumb. Why can't I just go straight up from the bottom? And the reason here is that, okay, so yes, you could go straight up from the bottom. You could just be like 0, 0 plus 1 is 1, 1 plus 1 is 2, 1 plus 2 is 3, etc. And so the Fibonacci series is not really good at, it's not really a good problem to show how powerful DP can be. It's just a good example as to like what method it uses to solve these problems. And in a sec, we'll talk about a DP algorithm that does take advantage of the use of DP. All right, so yes, okay, cool. But and then I don't think I emphasize this, but when I mean what he means here by overlapping subproblems, it means that you have to solve the same exact subproblem multiple times. All right. For example, here you had to solve what f of 2 is twice. All right. So instead of having to recalculate it twice, you just store its solution, and the next time you need it, you can just directly pull it out and use it. All right. So that's the idea here. Any questions so far? All right, cool. Awesome. You guys are all amazing computer scientists. So we can move on. Okay, yeah. So in summary, the idea is, oh wait, did I skip forward? Oh dear. Okay, yeah. Okay, yeah. So here's another example. I guess f of 5. Again, here notice that after you calculate all of these values, or sorry, after you calculate like f of 1, f of 0, f of 2, and you get to f of 3, you shouldn't need to recalculate all these ones in red when you need them to solve the problem. All right, so the idea here is you minimize the number of computations by storing the answer to similar problems. All right, cool. Yeah, so if that makes sense to everyone, let's move on how we're going to use this for sequence alignment. All right, so okay, first of all, what is sequence alignment? It is comparing two strands of DNA, their sequences, and seeing, I guess, like how similar they are. All right. So our goal is to find out how similar they are. And again, we obviously can tell that like if they have the same exact sequence, they're exactly the same. So they should be very similar. All right. But we have, say, maybe one base pair difference, and we want to know how we can get a computer to figure that out, and we want to know how to computer to figure out if two things are like completely off. All right, again, you may think, okay, it's super easy with just 4 nucleotides, but actual sequences, obviously, as you know, have like millions and millions of base pairs, and obviously we're not going to go through by hand to check every pair of nucleotides just to see if things are like that. And also one other thing is we have to account for is insertions and deletions. And usually that can also be a little bit difficult to see sometimes. All right, cool. Yeah. So why do we need to use sequence alignment? Well, I guess number one, there's a lot of places where if we know one DNA sequence from, say, species X is similar to another DNA sequence from species Y, we can make some judgment about whether those two sequences or what those two species are related, how closely related they are. And we can also use this to, if this sequence of this protein that we know what it does is similar to a protein that we don't know what it does, we can kind of infer that their functions are similar. All right. So here are a couple other applications. I'm not exactly sure what these last two are. If anyone knows, just let me know. Oh, ESTs, it's like express sequence tags or something. Basically, I guess if you map something to, you can map something to like a genome using this. Yeah. All right, cool. Yeah. So, okay. I think, I don't know what this last thing is, but I think it's kind of, it makes sense, right? So, okay. Yeah. So again, with longer sequences, it's a little bit more difficult, right? So we're going to kind of clarify our problem a little bit more, because I guess we want to like first define specifically what like the rules of our problem is before we actually do things. Okay. So the problem here is called edit distance. All right. So the question here is, what's the least number of mutations you need to convert one of the sequences to another? All right. And then the mutations we're going to call edits here. All right. So there are 3 types of edits. The first one is substitution. So replacing a C for a G here. The second one is called an insertion, and the third one's called a deletion. All right. So again, you guys know what these are, but in the computer science terms, they only look at the sequence, and they're going to define these as their 3 possible mutations. All right. If there's more, just say if there's a substitution and an insertion, that counts as 2 edits and not 1 edit. All right. So that we're going to try to find the minimum number of edits that 2 sequences have. All right, relative to each other. And okay, so again, we're going to assume that the minimum number of edits is just going to be like the distance of the distance of the 2 sequences. So this is called a Levenshtein distance. All right, cool. So how are we going to do this? All right. So first, we're going to make a grid. All right. So we're going to make a grid, and basically, what we're going to do is that each spot on the grid will represent the optimal edit distance between the sequence up to that point on both sides. All right. For example, all right, this part on the grid is going to represent the optimal sequence alignment of this part of this strand, and this part of this strand. All right. So can anyone tell me which part of the grid will tell me your final answer? Your final answer to the problem?
Bottom right corner.
Richard says bottom right corner. Why do you say bottom right corner? Oh. All right, cool. I think K is here, but
Yeah, like at that point you've aligned both sequences.
yes. Yes. All right, cool. Is K here actually? I thought I saw her send something in the chat, but maybe not. Okay, I guess not. So let's just keep going. Yeah, so exactly. So once you figure out, once you get here, then your problem's solved. All right. So okay. So that makes sense, I guess. Oh yeah.
She's
What?
in the attendees, she's here.
Oh, she's an attendee. Oh my gosh. Okay. All right, give me a sec. Why can't I see her? Oh, there it is. Oh, I see. Okay, cool. Do I have? I do not have power to do this. Oh crap. Okay.
Good morning.
Oh, hello.
So thank you for starting my talk while you're at it.
Yes. This is from 2 years ago. So.
Yes. I stole those slides.
All right. Wonderful then.
Yeah. So I can take over where you left off, because I'm also doing the DP lecture.
All right. Sounds good.
Well, you've already seen the first few slides of my presentation, but so I'm doing most of the bioinformatics lecture, and I'm actually a bioinformatician for a living. I have a PhD. I work for Thermo Fisher. I do microarray design, and I will be doing most of the talking. Giving also credit to co-authors David Wu and Kimmy Shen, who are the 2020 TAs who also made a fair portion of the slide deck. And so let's go back to some dynamic programming. And I'm pretty sure you saw these slides. What it is, I put in a little bit more explanation about what this is and why you would want this. The first instance of dynamic programming that most people would see is a Fibonacci sequence. And that is, it is what happens when you say, okay, what is the number that you get from adding the previous 2 numbers? And of course, you have to define the first 2 of the sequence, but then you get, okay, Fibonacci of 2 is Fibonacci of 1, Fibonacci of 0, 1. Then 1 plus 1 is 2, 2 plus 1 is 3. And this is a nice straightforward sequence that people will show elementary school students as a cool application of math counting. But the big idea that I'm putting this in here for is you have big problems that can break up into subproblems. And it's a solution that you can express very cleanly in terms of solutions to these smaller subproblems. And of course, if you wanted to compute a high number of Fibonacci, you would end up, if you wanted to do it naively, say, okay, what is Fibonacci of 5? You'd have to compute, got Fibonacci of 4, and then you have to compute all the way down. And then, oh, Fibonacci of 3, I've forgotten what that is. I have to go compute it all again. You don't want to do that. You want to cache the problem, the solutions, the problem, and just go back and look. Oh, I could look up in this table what my n minus 1 is. I can look up in this table what my n minus 2 is, and I get to do those computations only once and remember them, rather than trying to do it every time that I need that information. Okay, so dynamic programming seems simple enough. What does this have to do with sequence alignment? And the goal of sequence alignment is, if you have two sequences, how similar are they? And intuitively, okay, if you have these two, if you have two identical sequences, they're identical. If you have things that are off by 1, they're similar, they're not exactly the same. And then you can get sequences that just have nothing to do with one another. And you want to have a quantitative measure of how similar these things are. And this is useful for a lot of different reasons. You can find homologous genes and gene family members, things that look alike and probably act alike. You can get phylogeny, you can do species identification, you can find parts of proteins that might have a defined function, and you can assemble genomes that way. This does a lot of useful things for you. It's a very common task in bioinformatics. If you've got a toy sequence that's four bases long, it's very easy, but how similar are even these primer length things? You have to formalize the problem to deal with these increasingly complicated sequences. And I walked in when we were talking about edit distance. You got that part, but that is just the minimum number of edits that you need to take one string and make it into another. And that could be insertions, deletions, substitutions, whatever you need. Okay, and so your first step is to construct your grid. And this is your lookup table. And so your Fibonacci is one-dimensional. You've only got a one-dimensional string of numbers. This lookup table has 2 dimensions, and you need that because you could get your alignment from 3 different places. So if you want to know, what your next empty space is, you can get that empty space from any of 3 different places. You can extend your alignment without gaps to say, I want to take both my sequence from my A sequence and my B sequence. I want to take one letter from that, and just extend it, and get whatever bonuses or penalties are from that. Or I want to open up a gap. I want to take the first letter of my B sequence. I want to accept that, but I want to keep the empty space for my A sequence, or the other way around. I just want to accept the letter from the A sequence and not the B sequence. And then I can take that score and move it along. And of course, I want to take the best score I can get from those in order to give a value for that position. All right, so your first step in doing this is first you calculate your trivial cells. And the distance between any empty sequence and any non-empty sequence is just the length of the non-empty sequence. So all you're doing is, in order to go from empty to your first letter, your A, that's a netted distance of 1. All you have to do is add in that A. All you have to do is add in that T, and so on. And so each of these cells will go up 1 in edit distance. And the only portion where it starts to become interesting is how do you get to this next value? So you can say, okay, I'm starting from this alignment where I've taken my A, but I've not taken anything from my B sequence. And so I'm already starting at an edit distance of 1. I move forward, and then I am not taking anything from my A sequence, but I am taking a thing from my B sequence. That would be opening up another gap. It would be dash A for the first sequence and A dash for the other. So that would end up with, you'd have an edit distance of 2. In this metric, if you were opening up a gap, that is 1 point. And so say, okay, plus 1, and I've already started out at my at a score of 1. So in order to move forward from that position, it would be a total score of 2. Same thing for the one above. I start at 1, and then in order to move down, I need to add 1 as well. So I would be getting a score of 2 from that direction. However, if I simply extend, I'm starting out from an edit score of 0, and then I extend, and these things are a match. So I stay with my score of 0, and that's the one I want to keep. That's my best score. And then moving on, I can extend. I can open up a gap from here to here. That's gaining 1 point, but I'm starting out from 0 here. And so I'm starting my alignment with this AA, and then I'm choosing to open up a gap, and this T is not matching with anything. And that's a better option than moving forward from here, adding a point because these don't match, or moving forward from here, adding another point because these don't match. And we're doing the same sort of logic over here. In all of these cases, it is better to just move that gap along, and then you can start in on the next row. Okay, these don't match, so you'd have to go up. These would automatically go up a point. Your best option is from here to here. And for this cell, you're just proceeding along with your alignment. The first 2 bases in these match. You're keeping your distance of 0, and then like that previous one, you're just chugging along and opening the gap. All right, does that make sense? We're going to do another example, but it's one of those patience sort of things where it's easy to mess up rather than it's not rocket surgery. But it is possible to do it wrong under time pressure. And so in order to see what edits you did, what you do is you move backwards along the optimal path of the cells that you calculated. And so in this case, you are starting at the end of your alignment at this bottom right cell, and you are tracing back to what steps you had to take in order to get there. Okay. These last ones are a match. You're accepting that last letter from both sequences. In this case, you are not accepting your A. You are just accepting your B. So you are opening up a gap here in order to get to this portion where your T's match and your A's match. So you are aligning these first 2, and then this G is getting a gap, and then you're matching these A's as well. So the way you do that is you just step back, and then you trace the optimal path of the cells that got you from this lower right corner back to the origin.
I had a question.
Yeah.
So when you're creating these alignment matrices, is there a way to like determine the value in one grid based off of like what is immediate to its left, right, and like diagonally upwards, or like would you have to kind of just base it off of like what you're seeing in the sequence?
Well, I mean, it is based on the sequence. That's all the information you need. And so the point is that the only information you need is how good the alignment would be if you open up a gap in either of the sequences that you are aligning, or how good it would be if you continue on with the alignment as is and treat it as either a match or a substitution.
What does opening up a gap mean? Sorry.
So I think I have, it would be something like this, where you are trying to align your sequences and say, okay, these are not a match. These do not belong together. I'm just going to say there's no base here. It's a deletion over here.
Okay. So it's like in the algorithm, it's essentially like saying that at that spot there will be a deletion, just like considering how that would affect the alignment. Okay.
Yeah. So I've got kind of a more graphical representation of this. And so if you're trying to align these two sequences, your eye is telling you, okay, what this is, sequence 2 has this insert V, right? And so what you do is you start in this lower right and say, okay, these are a match. I'm going to accept the T on both sequences and proceed back here. And I am not going to just accept that V. I am instead going to go up and just say, okay, we have not gotten to this L yet in sequence 1. I'm going to open up a gap and therefore go up vertical. I will accept that V from sequence 2. And so I will proceed upwards on my sequence 2 map, but I will not proceed back towards the origin on my sequence 1 map. Instead, I will open up a gap, which allows me to, on the next step, right now I am on this square where I'm comparing the two L's. And then, okay, I can accept those. Then I move back here. I can accept the GG, and we are back to the origin. If that makes some more sense. And depending on what your alignment is, you can deal with mismatches, and you could take whatever penalty your alignment scheme has and say, all right, I am at V versus M. I'm going to accept that and not try to pick a fight over it, because I still have this area of better alignment up here. So I'm just going to accept it and move on. And I'm going to say that these two mismatched sequences do in fact belong together. That's the best solution. Rather than trying to say that, oh, it's mapping onto this L or something else, or there's been some insertions and deletions in here. You can just accept it and move on. But in some cases, if you have an area of better alignment, the best answer is just to say, oh, there must be a gap here. I'm just going to open up that gap and proceed. And so this is what opening up the gap looks like. If you have your sequence 1 with the gap, you move up, and so you accept your sequence 2 base, but not your sequence 1. And this is what it looks like if you have a gap in sequence 2. You move sideways. You accept your sequence 1 base, but you do not accept your sequence 2 base. All right. So you saw edit distance. That's a nice simple metric. It is not perhaps the most useful one biologically. And so here's a somewhat better representation of the situation biologically. We want to penalize having a gap. And we want that to be worse than having a mismatch. It's more likely that you get a substitution than an indel. And so here is a also fairly straightforward scoring scheme. And this is just, you can set this to whatever you want, but this is the also relatively straightforward way to do it. So let's say we have a scoring where each match gets you 2 points, a mismatch is neutral, and a gap penalizes you -1. And you can just intuitively score these sorts of mappings. So what if you try to align these by eye? Say, okay, you got your 2s, and you could see that maybe this this ungapped alignment is better than what you could try to get if you tried to shift. But you could do this more formally, and you would have to do that for a bigger problem. All right, so let's use this scoring system and try to score these 2 sequences. So we begin, we're starting at the origin. The first thing that we do is we fill in the trivial sequences. There we go. Because each of these, they're no longer just adding a point. They're now you're now getting your penalty, and you're losing a point here. Every time you open a gap, it's the same thing. Above, you're just opening up 1 gap per sequence where you proceed. And now you get into the interesting portion who say, okay, I'm going to, what happens if I open up another gap heading down? Then I would get 1 more gap penalty, and I'd be at -2. What happens if I extend from the origin? Hey, these match. It's actually +2, so I get +2. And then opening up a gap is worse. So obviously, I'm going to take the extend option. I'm going to end up with a score of 2 for this. That's not good. That's not good. That is exactly what I want. So there's my 2. And here again, what happens if I extend down? I'm already starting with a fairly good score, so I'll just lose a point. Coming across, trying to extend after I've done my gap. You have that sequence, but that's worse. So that's what that is. 2 gaps and then adding in a third. So my best option is to say, okay, these A's match, but this T and the A are going to get a gap here. And I would accept my 1 as my best score. And you can fill in the rest of the table using the same method. If you remember, now we're going to trace this back. We're going to find the path that we took starting at the end and working backwards. And so we're starting here. Where did I get this number from? I got it from extending. I got it from here. Where did I get this number from? I got it from opening a gap. Where did I get this from? Extend, extend. So my final solution here is going to be this, where I've opened up my gap here, where I've accepted my G but not my T here. I've left my T for future endeavors. And this is the final alignment. All right. And so, of course, this is biology. There are some more complications in here. It is possible to align multiple sequences. I am simply going to say that that is possible, and it uses the same idea conceptually. You're just generalizing it to n dimensions. But there's also the problem where you don't necessarily know the starting and end points of your alignment, right? It is a very convenient thing to do is say, okay, start here, end here, and make that happen as best you can. Often you will have just two sequences, and somewhere in there might be a match, and you have to figure out where it is. And so how do you do that with this kind of approach? What you do is you take this generalization, they call it Smith-Waterman, and the idea is you're not looking for your end position anymore. You're looking for your best value. So okay, here is where I am furthest above the background, and I'm not necessarily at the end of my sequence, but that's fine. And then starting from that best value, I'm going to trace back just as I did in Needleman-Wunsch, and I'm going to figure out where that value came from. So I'm extending, I'm matching my C's, I'm extending, I'm matching my A's. I am going to go vertically, I'm going to accept my G, but I'm not going to accept that T. I'm going to keep those T's and then accept both of those T's, and so on, until I get to a point where there is no longer any value to this alignment. I have reverted back to noise. And so I am not going to consider these two values aligned. And so only this above ground section here is considered as locally aligned in this case. So that is how you use this approach when you don't know the starts and stops.
Also, just checking, this approach uses different scoring values as well, right?
Yeah.
Right.
The scoring function for these are fairly arbitrary. You can use, they have some different ones they would use for proteins, obviously. Proteins are the ones where it gets the most diverse. With nucleotides, only 4 bases, you can't get that creative, but amino acids, you can take into account the biochemical properties of it. Is this a conservative or a radical substitution? And so you can get a whole complicated scoring matrix, which I'm going to get into later. Not yet.
And regarding the scoring for the Smith-Waterman, is there like a, I guess, intuitive reasoning why the scoring here is different than that of the one we looked at earlier? Or is it?
It's largely convention, but it just makes it easier for the background to be 0, because this particular scoring function, you get negative values. All right. So I'm going to give you 5 minutes to try it yourself. Here's a slightly different sequence. Just grab your pen and paper and work through it and make sure you can. I'll give you a few minutes to think about that. All right. Now that you've had long enough for me to get my caffeine, let's bring up some answers. All right. hope you got some of these. It's the same approach. And how do you get to your final answer sequence? Starting here, and you are going back to here. Note that you're going back to where you got your answer from. You're not necessarily going back to the best sequence. And so you are accepting these A's, both of them. You're not necessarily opening up a gap, even though this alignment here is surprisingly not that bad. And then from here, you have a choice. Which one would you like to accept? And so you can extend even at a mismatch, keep both of them, or you can open up a gap and then be back here. And these are equivalent, and so you have these equivalent alignment paths. If you extend, then you're going to end up opening up a gap elsewhere, because you're going to end up. So if you extend back, you're going to go from here. These are misaligned, and then you're going to have to open up your gap. Once you get here, you're going to have to get back to the origin. And if you decide to open up your gap here, and then you will just step back in extension back to the origin here. Obviously, you're not going to open up a gap right here where you have your one base that matches up, but you can have ambiguity. All right, are we good here? All right, I'm seeing nods. It's a good thing, especially this early in the morning. Everyone likes dynamic programming first thing in the morning. Past the caffeine. All right. Part 2.1, Motif. And what is a motif? It is a sequence. It is a sequence of DNA that has a form, a stereotypical form that you can look for. And so the reason that motifs come up is they are commonly transcription factor binding sites. That's not the only thing they do, but that's a very common thing. You're looking for a short sequence with these typical properties, not necessarily an identical sequence like a restriction enzyme site, but a family of sequences that are related and they look similar enough that this transcription factor can bind to them. And these are useful for if you're trying to study gene regulation, you want to know what things this transcription factor can affect. This is something that I probably don't have to go deep into, but transcription factors, these are proteins that are encoded, and they recognize these specific DNA sequences. They recruit the transcription assembly in order to make this gene do, or if it's an activator, recruits the transcription assembly, make it go. So repressor recruits repressive assemblies, and it brings in the repressive factors and modifies that area of the genome to say, nope, I don't want to be able to transcribe this anymore. And these things are operating based on sequence recognition, and they don't necessarily have an exact match sequence. They recognize a family of sequences. And a way to represent the preferences of a transcription factor is a motif. And you'll see these kind of diagrams it is a sequence, and the size of these letters represents how strong that preference is at that position in the binding site. And for this particular transcription factor, it has a region in the middle and at the edges of its binding region where it doesn't have a strong preference for what the base is at that position. But you can see at the 5 position, it pretty much has to be an A. And at this 12 position, it pretty much has to be a T. And some of the other sequences, you have your choice of 2 bases that you can get away with, but really not all 4. And so that's what I'm talking about, where it has a preference, but it is not just, here's the sequence, Control F. And so you have to represent it as this motif, this family, this probability cloud of is this something that it was like. So how do you get, where's the data? Where's the raw data for figuring out your motif? And so one way that you can do this is you can do cross-linking experiments where you have your transcription factor, and then you treat it with formaldehyde so that all the proteins stick to where they are, and then you take an antibody and you collect everything on that transcription factor using an antibody to it, and then you sequence what has been pulled down with it. You can do a similar conceptually sort of experiment with some in vitro binding and some amplification, where what you do is you incubate a library of sequences with your transcription factor that is bound something, and so you are capturing the sequences that are able to bind to your transcription factor, and then you do some amplification of that, and that is your new pool. And so you select for the things that want to bind to your transcription factor of interest. But the general idea in the end is you will get out a panel of sequences that are associated with this transcription factor. All right, so you've done your experiments, and now you have a bunch of N-mers. And now you want to know what the motif is. What is the commonality between all of these? All right, so here's your toy example. Let's say we did our experiment, we came out with these 4 sequences. And what we do is say, okay, we have this grid, and here is all 4 possible bases that it could be. And here is the position. And we look and we count. Okay, in this first position, what bases do we have? We have 4 A's and we have nothing else. That's what we got here. What about the second position? We have an A, T, a G, and a C. We've got 1 of each. And so that's what we put in our motif table. Third position, now we're back to having all A's. Same thing with the fourth position. Then the fifth position, say, okay, we have some A's and we have some T's. We don't have anything else. That's 2 of each here and none of these. Counting. And And so now we convert these things into probabilities. So the probability of having an A in the first, third, and fourth position is 1, 100%. Whereas the probability of having an A in the second position is just a quarter. That is, yeah, just divide it by the total. All right. And so I talked a little bit about uncertainty and entropy. And so what the intuition here is, you have these probabilities, but what that indicates is that some of these positions are more informative than others. And so if you saw something that is not an A in the first, third, or fourth position, you would have a pretty good idea that that's not part of your motif, right? Whereas if you saw whatever in the second position, it would tell you nothing, because anything can go there, it's fine. And this is obvious in a toy example, but if you're going to do this at scale, you need to formalize it. And so we have this notion of entropy, or how informative or how surprising it is. And so this is this is going back to this idea from communication theory, where you have your entropy is the sum of the probability times the log of the probability. And what you end up getting is, so for this row 1, you have this entropy of 0, because one of them is 1 log 1 to log 2. But intuitively, there is no disorder here. It's A or bust. You don't have any randomness to contend with. Whereas position 2, it has perfect randomness. It has an equal representation of everything. And you end up, in this case, with 2 bits of entropy. And then your position 5 is somewhere in the middle, 50-50. You're randomizing between 2 bases, and you end up with 1 bit of entropy. And the big idea is that the less entropy, the more informative it is, and the more surprised you would be to see something else. And so the amount of information you have, or the certainty about the binding preferences that you have in each column, that is, it's the converse of the entropies. And so your max entropy is 2, and then the information column is your max minus your observed entropy. And so in order to convert this into information content, that's all you're doing. That's 2 minus your observed entropy. And so in this first column, where your entropy is 0, there are no surprises. Your information content is the maximum available, you get 2. And in the second column, where there's no information, you have maximum entropy. Yes, I have my furry little entropy generator trying to eat my keyboard. No. All right. And so now we can get our probability matrix and reweight it by information content. We have the idea that some columns are more important than others. And so now we can generate our position weight matrix. And this is a thing that we can use in order to score potential motifs, to see how well they match. And so here's how you get your probability matrix. And so you are multiplying each column by its information content. And so for your first, third, and fourth columns, you have your full information content of 2 times your full probability that lives in your A position. And so these positions get the maximum score of 2. And for column 2, you have no information content, so nothing that you do here matters. Nothing that you do here will get any points. And here you have your half dose of information. You get your 1 bit of information, and then that is shared over these 2 positions. Here is your logo diagram representation, where you have your 3 strong A's, your 50-50s, and your nothings. All right, so how do you use this to predict a transcription factor binding site? All right, so we're going to use this position weight matrix, and we are going to scan this thing across the entire genome to look for matches. But we're doing this by hand, so we have our little toy genome over here, and say, okay, how well does this first 5-mer match? Okay, that matches. That I don't care. Oh, that doesn't match. And so you can get your score. Say, okay, we have we're getting credit for this A. We'll get our 2. Nothing for this. Nothing for this G, or for this C, or for this G. And so we're scanning across the genome, and then we can move on to this one. Okay, here's your first position, a 0, and so on and so forth. So you're just throwing this thing at the genome and say, okay, do any of these look like my transcription factor motif here? So let's calculate what that looks like for this individual 5-mer here. And so in order to get your match score, you would need the match score is on a 0 to 1 scale, and so in order to normalize it, you get your maximum match score. And so the best that you could possibly do here, you can get a score of 6.5, right? You can collect all your 2s here, and then you have your choice of these 0.5s here. And then in order to get your realized match score, you have to go through each of these and figure out, okay, so this first one is A, it matches, you get 2 points for that. Second one, T, you get 0 points. Third, A, you get your 2 points. Fourth, C, no points. Fifth, C, no points for that. And so you get your 4 points for 1 and 3, and then you divide that by your maximum score, like so.
When it comes to these match scores, is there like some threshold of which you would say this is probably a TF binding motif? Like, or is this kind of just relative?
It's relative. It depends on the transcription factor.
But does each transcription factor like have a threshold value?
So partial binding is a thing, or weak binding is a thing. And so it is not necessarily the case that it's not, binding is not always binary. It can weakly associate and fall off easily, and sometimes that is the behavior that you want, biologically speaking.
So like, would it be like, how would you interpret this match score that's calculated here?
It would make most sense looking at it in a genomic context, and say, okay, this is one of you'd get your distribution of match scores, and ideally you'd say, okay, you have this population that has a match score of 0.9, and then the rest of the genome is just on the tail, and then you have your small population that matches really well. And so you could look at those and say, okay, these are more likely to be something that I am interested in. All right. And so one further wrinkle on this is you don't always have this nicely defined area. Say, okay, here's where it starts and where it stops. That is a luxury we don't always have. And so here is how this is dealt with in the real world, where you have your pull down experiments and you have your oligo, where there is a motif in there somewhere, but you don't necessarily know where it is. It's just enriched. And so you use sampling. And what you do is you start with a random position in your sequences that you believe have a motif in there somewhere. You just take a chunk of it, and then you build your weight matrix. Say, okay, if these are actually motifs, what does my weight matrix look like? And then you take your sequence at random, and then you score the possible sites in the sequence using your candidate weight matrix, which you are hoping is slightly better than nothing. And you're looking for, okay, if this weight matrix is slightly better than nothing because I've enriched for my motif, what part of this target sequence is really my motif? And you take a new site, and you want to make it proportionally more likely that you pick a good target. You overweight the ones that score highly versus the ones that don't score. Like all evolutionary processes, you want to take most of your probability from the good ones, but you don't want to get stuck. You know, if you've chosen your beginning sequences kind of poorly, and you refuse to take anything but the best sequence, you will never get out of that local optimum. Whereas if you take too many sequences from the bad that just don't look like much, you can be so open-minded that your brain falls out. You know, you have to live in the happy medium where you're mostly biasing towards the good sequences, but you are also exploring your space. So you're sampling proportional to the likelihood that this is actually part of your motif in training.
I have a question. So this graphic kind of looks like this new site is being added into that sample. Does that mean that the weight matrix is being recalculated after every single iteration?
Yep. You're going through the loop, and you do that again and again and again, and you do that until it converges. You are not finding new sites, or your weight matrix isn't changing. And so that's how you evolve your way towards a motif, if you don't know exactly where it starts and stops. You have your random position. This is done by chance, but this is presumably something that you got out of a SELEX or an enrichment experiment. So you know somewhere in here, there's probably a motif. You build your initial weight matrix based on assuming that these things are the motif, are the binding site. You take a sequence, and then you figure out, okay, where along this this sequence is likely to be a good motif that matches everything else in my pile. If you are a good match for what else is in the pile, you can go into the pile, we'll recompute the matrix, and then we'll do it again. We'll do it again, we'll do it again until it converges.
Sorry, so this initial random sample that you have, is this within a single sequence that you believe has motifs? Or are you taking like multiple oligomers that you've somehow generated through like some lab?
Generally, you would take one site, and so this would be your 15 base chunk of whatever you got out of your experiment. Might be like a 15 base chunk of 100 more.
So like the initial, so you have a random sample of sequences and then you take a random one of those to cycle through?
So your inputs are not going to be random, ideally.
Okay.
You have done what you can in order to enrich biologically. So you've done a pull down experiment and you've sequenced what you pulled down. You've done several rounds of antibody pull down followed by amplification. And so you have reason to believe that much of this is motif. And if you seed it with garbage, it's going to take forever to converge, and it will probably not converge to something meaningful. That's garbage in, garbage out, right? However, if you have something where these proportions are more or less right, where you have this motif that is making up maybe 15–20% of your input and you don't know what it is, then you can get enough information in your initial sample that you can start to go on to the right path and fully identify that motif. But you have to do the work up front in order to clean your data, and that is an experimental thing. Obviously you can't just throw in the entire genome and say, find me a transcription factor binding site. I mean, it'll probably find you like a sign element or some transposon. It's not very informative. I find you a centromere.
All right, thank you.
I find you a cat. Hi, kitty. Miss you. You like Gibbs sampling. All right. Next, phylogenetic trees. All right, what's a phylogenetic tree? And so this is the tree of life. You'll see these often, and these are a representation of ancestry. And you're trying to infer the ancestry of a set of living creatures based on their traits. And you can use all sorts of traits depending on your application. You can do it old school, and you could look at them and try to look at their morphology and classify based on morphology and say, okay, these ones over here have an amniotic egg and these do not. Probably these are related. You can do it new school, and you can look at the sequences. But these are used for a lot of different things. It's evolutionary history. It's trying to identify gene families and trying to figure out where virus came from and where it spreads. It's a very common task for bioinformatics. And so first we need an idea of evolutionary distance. And this is something that you would primarily look at for sequences. One measure of distance is just the observed number of substitutions per site. That's a nice, easy thing. That's edit distance we've talked about. Something to be aware of if you are using that sort of distance metric, is that it is only linear with evolutionary distances when things are closely related, right? So if you have a short distance, any distance between any substitutions, any differences between the sequences, that is likely to be a one-for-one thing. Each difference that you see represents a single mutation. And so the number of mutations is linear with the distance or the amount of time it has been since these things had a common ancestor. As you proceed further and further into the phenotypic distance and these things get less and less similar, you start to get instances where a single site has mutated multiple times. And so you can't just keep proceeding into infinite phenotypic distance. It's like pure randomness. You're going to match a quarter of the time just by chance. And so you'll get this asymptote, whereas as evolutionary distance increases, your phenotypic distance increases more and more slowly. And so there is an equation for it, where you can take your phenotypic distance and plug that in and get an estimate of your true evolutionary distance, which goes exponentially up the closer you get to the closer you get to that 0.75 randomness mark. But if you are living in this fairly safe regime where things are not that different, there may be 20% difference, then you can treat these things as identical. And most of the things that it would make you do by hand, you can live in this range where you can treat phenotypic distance as evolutionary distance. That is something to be aware of. As you get into larger separations, that is no longer true. All right, so what are some methods for creating these sorts of evolutionary trees? Some of them are simple enough to do by hand, and that is something that people would like to inflict on you in the practical section. And so you will need to know how to do that. The most common methods are these UPGMA and its weighted cousin, the UPGMA. And I will show you how to do that. The idea is you start with an alignment, which we've talked about, and you generate a matrix of the distances between these different sequences, and then you're using that as your raw material to make a tree. I'll show you how that works. And so what this is this is a pairwise calculation. How different are these sequences? So first thing you'll notice, obviously, these things have zero difference from each other, from themselves. And so sequence A has zero difference from sequence A, and so on. So it's a hollow matrix. You can't have a negative distance. You can't have a negative difference. And it's symmetric. The difference between A and B is the same as the difference between B and A. And so this calculation can be something straightforward, like you can have an edit distance of 2 between the two of these. You can have a phenotypic difference. You have some scoring function, but for this to work, all you really need to know is that if you have a distance of 2, those are more closely related than if you have a distance of 4. That is the only property that you need to have. And so underlying this distance matrix, there's a tree. And you want to find that tree. And so what does that look like? So if you have this tree, this truth underlying your distance matrix, so what is your distance between A and B? You have your 3 to get to your common ancestor here, your 4 to get to your common ancestor here. You have a difference of 7 between A and B. What about A and C? 5, 10, 12 to get all the way over here. That's a distance of 12. And here you have 5, 8, and 8 in order to get here. So usually you will not know this tree. All that you can see are the leaves, and you can see how different they are from each other. And then you have your distance matrix. And what you want to do is calculate this tree over here. And that's the thing you can do. And so what you want to do is come up with an algorithm for doing that. What UPGMA and WPGMA are, these are clustering methods. And they're trying to agglomerate these related things. They're trying to figure out, okay, what of these individuals or clusters, which of these are the most closely related? Let me agglomerate them, stick them together, and make those into a bigger cluster. Say, okay, in this case, say 1 and 2, these are the most closely related. Let's stick them together. That's a super cluster. And then 4 and 5, those are the most closely related. Let's stick them together. And then we can look around at our options and say, okay, 3 is of the remaining options, this 3 individual and this 4, 5 cluster, those are the most closely related. So we'll stick those together. And so the difference between them, UPGMA is looking at the average pairwise distance between clusters. And so if you want to find the difference between, or the distance between these, you're taking into account all of the connections between the individuals in that cluster and not the average in that cluster. And the average is where you get into WPGMA. So here is an example of how you do this. All right, so we have a matrix. Here's our distance matrix. And let's say, for the sake of this example, we've already clustered A and B. They have no difference from each other. And so we're giving you that for free. A and B, they have already been combined into a 2 individual cluster with 0 distance. How do you decide where the next cluster merger is gonna come from? All right, so what you do is you look and you say, all right, where's the lowest difference? And in that case, you have your C, and that is the distance between your A and B merge cluster and your C. That is the lowest available. And so you want to merge these things together into one big happy cluster, this thing here. And so how do you recompute the distance matrix here? So, okay, so for UPGMA, you are taking into account all of you're calculating the average, but you are calculating the average of all individuals. So you start with All right. What is the distance from this AB cluster to D? And so you are taking that into account, and you are taking into account what is the difference between C, which is your other component of the cluster, and D. All right, so the distance between A and B and D, you can just look it up on this chart. It is 9. However, in this case, you have to count that twice because there are 2 individuals in the AB cluster and they have an average distance of 9. And so have what is your distance score contribution from those 2 individuals? You have an average of 9, 2 individuals. And then you have your 1 individual in cluster C, and the C and the D have a distance, CD, that's 6. And there's only 1 of them. So what is the average distance of these 3 individuals? That's 2 times 9 plus 1 times 6, and then 3 individuals, 24/3, that's 8. We are going to be working more examples. The difference between, so the other approach that you can take, what if you don't care about individuals, you just care about clusters? And so you can take the same example, and what if you just wanted to look at the average and not care how many individuals are in a cluster? All right, so you start in the same position where, okay, I'm starting with my ABs merged, and then I have my C and my D floating around. I don't know which one I want to pick, but I can look and say, all right, the difference between my AB cluster and my C cluster, that's the minimum. So I'm going to merge those together. This is one big happy cluster, and now I need to recompute my distance matrix. And so how do I do that? I am once again looking for the average, but I am now only considering the average of the cluster differences rather than the average of the individuals. And so what is the difference between my AB cluster and my D? That is 9, and that's just a simple distance. That is 1 difference between clusters. And then what is my difference between C and D? Look that up as well. That is 6. And so when I want to go over here and recompute what my difference is from this ABC mega cluster to D, I just take the average of this cluster difference and this cluster difference. So that's 2 things this time, 2 units to average between. And here's my individual contributions. And then I end up with that average. All right, so again, the difference between these 2 methods is in this, it's a somewhat confusing nomenclature, because what is unweighted in here is the individuals. You're considering each of these individuals separately. And so what ends up happening in the math is that you have to weight the size of the clusters. Whereas in order to the weighted average of the individuals, you are not considering, in the form of the math, you're not considering the individuals, you're just considering the average of the clusters, and so you end up not having the term for the number of individuals in the equation. But that's why they call it. All right, practice time. You're going to need practice time. And so let's try this. I stole this example from Wikipedia. It is the difference between some ribosomal RNA and 5 species of bacteria. This is real data, but But for our purposes, these are 5 strains of bacteria, A through E, and these are the differences between them. So I'll give you a few minutes to think through what you would do for your first cluster step.
Hey Kate, real quick. Actually, do you think we can like somehow like take a break sometime soon, or maybe have them like try to solve this like during a break and take like a longer 15-minute break or something like that?
Okay. So this is a little practice session. So how about I stop talking and come back at 10:45, and you can think about this. If I can send you the Wikipedia page as well, because I stole this example directly from Wikipedia and it goes through it.
Shouldn't, like, I had a question about like ED and EE. Why are the distances different? Shouldn't this be symmetric? From E and D, it says distance is 6, but from D to E, it says it's 43.
That is a typo. I'm going to fix that real quick.
Maybe using 43 while we're like doing this practice.
Yeah, that's supposed to be a 43. And like I said, I stole this from Wikipedia. So if you want to see it step through, I have a cheat sheet for it as well. All right, first step, find the smallest number, the difference between A and B. And we're going to merge A and B into a single cluster. And you would do that by looking at what's the difference between A and C, what's the difference between B and C. And so you would take the average of the distance between these, and then you would get your new distance measurement for what the difference between the AB cluster and C is. And so that's the average of 21 and 30 here. And then you would go along and update these using the same principles. And then in this case, what is the new lowest number? Be looking at this 22. And then so you would be merging in this E, but this is WPGMA. So you are still using the simple divide by 2 average of what was in the AB distance and what was in the E distance to each of your still existing clusters. And then you would end up with this final result. And UPGMA is totally the same for the first step because nothing has been merged yet. But once you have been merged, then you have to start taking into account that there are 2 of these things in here. So if you want to know, and so you can say, again, this is the smallest distance between clusters. So if you want to merge these 2 things, the difference between A, B, and C is this, but you have to take into account that this distance represents 2 different objects. So you multiply that by 2. So that's your distance between the AB component of that cluster and C. And then, okay, what's the difference between E and C? And so that only counts once because E is 1 individual. And so the average would be 2 times 25 times 5 plus 39 divided by 3 total. And that's how you would get the difference between the combined ABE cluster and C. And so that's how you would get 30 here instead. And you would keep doing that, and you would end up with the same tree with somewhat different estimates of the difference between them. Yes, and I did get a request to go back to some entropy tables real quick. And so a somewhat more complicated example, what happens if you have three things that have not been there, that where there is some density? And so what if there was a sixth position where half the time it's a C, and then the rest could be either A or T, and then you're not seeing any G's. You can do this the same way. You go through and you say, okay, what is the entropy at each of these positions? And so you have, what is your uncertainty here? So it turns out that your, if you have a probability of 0.25 at your position A, and then you would end up with a weight of 0.5 here, and you would also end up with a weight of 0.5 here and a weight of 0.5 here, and so you would end up with this position is, it's not that informative. You would end up with an entropy of 1.5, because each of these positions would contribute 0.5 to your entropy. And then you would get, that would leave 0.5 for your leftover information content. And then so you would go and you would multiply each of these things by 0.5 in order to get your position weight matrix. So you can do that. So that's how you would handle the intermediate case. So one thing to know about this method is that it assumes that the molecular clock is constant over time. So mutation rate, it's happening at a defined rate, it's not changing, and that is an assumption. It's very much an assumption, and it is often not true. So if you have stabilizing selection going on, see over here, that you're just not changing things very much. In the case of these populations 2 and 3, then you could fool this algorithm into thinking that your 2 and 3 are the most recent divergence, whereas they're an older population, and they are just not, they have just not diverged very much. And so there are alternate ways of looking at this. And so in order to avoid this constant molecular clock assumption, you'd use some other neighbor methods. The oldest and simplest is so the score of difference between your clusters. You can use nearest neighbors, where your score is how close the closest neighbors that are not in the same cluster are. You can use farthest neighbors, which is, okay, what is the most divergent example in these two clusters? And so those are the older school methods that you might do by hand. There are newer school methods that are just kind of impossible. The one name that you might see mentioned is neighbor joining, where the idea is that you are trying to start with this star and then figure out where to join these clusters such that the weight of the edges in the finest tree is minimum. This was a very popular method in the earlier days of computer because it is fast and computationally efficient. It's not so robust to noisy data. It has a tendency to give you negative edge weights if your data is not perfect. And so there are newer methods. This is where you get these kind of star diagrams from, and it is trying to minimize the total edge weight. And that's really all you need to know about that one. All right, so we've talked about these distance-based methods. And you can also look at character-based methods. So you're looking at, in these cases, you're trying to look at parsimony. You're trying to look at what is the minimum number of evolutionary steps do I need to get to a place. And these character traits can be a lot of things. They can be morphological traits, as you see here. The presence or absence of these life history traits, or so it's like hair or mammary glands. If you want to classify animals and things that might be mammal adjacent, or they can be something like amino acids in a protein. And so you will get this character matrix reflecting the presence or absence of a trait. And then you can use that to make a maximum parsimony tree. And so the optimal tree is the one that requires the least number of mutations to get there, right? That's Occam's razor. The simplest answer wins. And you're trying to figure out what is the tree that requires the fewest mutations. You say, all right, so if you have this set of sequences, how do you get there? It probably makes more sense if this ATCC sequence is off on its own branch, rather than having to start with a common ancestor that did not have a T in the second position, and then it independently acquired it twice. Okay, and so how do you solve, or how do you score a tree for parsimony? And we're going to do a toy problem with this. It's called the Fitch algorithm. And so what you do in order to count parsimony, you start with the base of this tree. This is your assumed tree. And at each step, you want to project back up to your vertex and say that, okay, what is this ancestor here? And in this case, you have two different traits. So you don't know what the ancestral trait is, and so you say, okay, I don't know. I'm going to say the ancestor is one of these things here. And so you take, you don't have a common thread, so you have to conserve both of them. Take the union up here, and you're doing the same here. It's either a T or an A. You project that up to the vertex. It's either an A or a G. You project that up to the vertex. Here is where it starts to get interesting. And say, okay, the common ancestor here, I don't know what it is. And this is a G. So I'm going to say, all right, the intersection of all the things that it could possibly be is no longer empty. And so I have a shared thing that this could be. And so I'm going to guess that my common ancestor up here was G specifically, because that is the most sensible solution for having one branch that is G and another branch that contains G. it is most likely that this A is a mutation, and I'm going to keep the G at this vertex here. And we're doing the same thing here, projecting these back up to the vertex. Both of these children nodes include a T. And so it is going to be likely that this common ancestor here was a T. And now, once again, we don't know what the common ancestor is here, so we have to go and keep them both for now. All right, so you filled up your tree, and now you could go down and you can calculate the number of mutations that you had to go through to get there. And so one thing this doesn't mention, if you don't know which one is the common ancestor, you just have to guess. It works out the same either way. Let's say we've guessed the common ancestor is G. And then here is mutation number 1 in order to get here. And then here's mutation number 2 in order to turn it back into a G. Here's mutation number 3. And it's still a G, it's still a G. Oh, nope, that's mutation number 4. And so we would have a parsimony score of 4 on this tree. All right. If you're going to do that on a lot of trees, it gets ugly. It gets ugly real quick. And so obviously you are not going to be doing this by hand for very large problems. The only thing you really need to know is that this goes on combinatorial growth, and you have to start using approximation methods in order to make this work, because you can't just search every single tree in order to find the best one. And there are various methods for doing that. Some of the things that can come up if you're doing these methods, you have to be somewhat careful of ignoring convergent evolution. And so you might see if the true case happened where A and B are in similar environments, and they picked up a couple of similar phenotypes in order to survive in that environment, this might look like these are more mutations, and you would want to try to put those closer to the root of your tree, but that might not necessarily be what's going on. So especially if you're using phenotypes, ideally you would go with less correlated phenotypes. That's the thing to be aware of. And in general, there's a trade-off between how likely it is to find the very best solution and how much computer you are willing to throw at it. The more supercomputer you have, you will start to round up these corner cases a little bit better. But obviously, there's a limit to what can be done practically. All right. Oh, we're making good time. I'm going to give you a 5-minute let that settle in break and think of any questions. That makes sense before we head on to everything else and to a BLAST practical exercise. Get back to everything else. This is more of a grab bag of odds and ends, less mathy, and then a BLAST exercise. The exercise is actually taken from a previous practical, a little bit too evil. Somebody got it. Ideally 4 persons would get it, but you get it as a practice problem instead of me trying to run it back. All right, let's go. So part 3, everything else. Some more big ideas in bioinformatics that you should probably be aware of. GWAS, genome-wide association studies. It's a very common thing to be doing with your genomic information. Another common thing to be doing is gene ontology. It's looking at annotations for your genes of interest, and some discussion of why protein folding is hard, and then when you don't really want to get into all of that alignment stuff, you want some heuristics. Well, we're going to talk about the most common accepted way to do local alignments, not in the purest way that we did and we worked through by hand, but in this fast heuristic way. It's BLAST. Everybody uses it and loves it, and we're going to do a tutorial on that. Okay, so on GWAS. So this is something that you will see all the time in genetic studies. This particular data happens to be associations for sodium concentration in Arabidopsis thaliana, but you'll see these sorts of plots all the time. They're trying to ask the question, what genotypes are associated with a particular phenotype? And this style of plot, it's called a Manhattan plot. The idea is that it looks like a city skyline. It has these occasional skyscrapers. And what the axes are, the x-axis is distance along the genome. In this case, it's colored to indicate the different chromosomes. And then the y-axis is the p-value of that hit. You're looking at, does this population have a disease or not? Does it have high sodium or does it not? And you're looking for individual genetic markers, individual SNPs that might be enriched in one phenotype versus the other. And so you're puttering along your genome, and you were seeing the stuff that comes above this Bioinformatics line, where this is where you would expect, if all of this was noise, you would expect these things to be below that line, even if you take an entire genome's worth of samples. If you sample something 1 million times, just by chance, you're going to get some pretty strong associations. But this line says, okay, here is the cutoff where you would expect, and you would expect your noise to be below that cutoff, even though you have taken all of these samples, and things above that line are deemed to be significant hits. And so, what are these hits? These hits are generally marker SNPs. They are not necessarily the thing that is causing it. And the way that works is it's linkage disequilibrium. So you have your genome, and it is a long chain of SNPs, and they're stuck together. And they have an association that persists from generation to generation because they are physically attached to one another. They can recombine, but if they are close, it is a slow process and it takes place over generations. And so if you have a genome-wide study looking for common SNPs, you might have your data on this genotype SNP over here that is close to it. And that marker is in linkage disequilibrium, which means it is physically attached to this thing here, and it is only going to randomize over many generations. And so it is in linkage disequilibrium with what this unobserved causal SNP might be. And so it's this causal SNP that is actually doing the phenotype. It's actually having the effect that you observe. And so you can see, oh, this SNP that you're genotyping has an association with the phenotype. But that is an indirect association. And what's really happening is the genotype SNP is attached to this causative SNP, and the causative SNP is doing the thing with the phenotype.
I had a question about this. So if this is in linkage disequilibrium, would like the causative gene itself not show up on the GWAS? Aren't they inherited together?
So if the causative SNP is being genotyped, then yeah, it'll probably show up. It'll in fact be the strongest hit. But in many cases, you won't have a genotype for that. It might be a rare mutation that just didn't make it onto your SNP panel. This is more relevant for you're doing like microarray type studies where you only get a few 100,000 data points. You don't necessarily have full sequence data. If you do, then you can treat everything as a data point. So you're going to have your causative gene in there somewhere. And so you don't need to think about this as much. But often these studies are being done on these platforms where you don't have the full data. Even if you have something like full exome, you might have an intronic SNP and you get an association, or you might have a transcription factor somewhere off screen, some sort of regulatory SNP. And you'd have to say, okay, there's an association here. And just because these particular mutations are coming up with a signal, that does not necessarily mean that it is that thing specifically. It's just telling me that it's in the neighborhood. Yeah, unless you really have great NGS and you are absolutely sure that you are seeing everything. Infinite money, infinite data, it's great, but we don't usually get that. And so here's some graphic explanation of linkage disequilibrium. You have to wait many generations for recombination to start scrambling these things. You have these patches of inheritance from the parents in this initial generation, and as you get a couple of recombination events per generation, slowly this starts to decay, but it takes a while. All right, so sometimes sometimes traits are simple Mendelian, right? Where you have this very specific mutation that causes this very specific phenotype, and you can home in on it on your GWAS, and it lights up like Christmas, and everything is easy. Get that. You'll get complex polygenic traits. You get things like height or weight or intelligence, or cancer risk, where there are hundreds or thousands of causative SNPs or causative variants, but they all individually have a weak effect. You would still want to do something with that data. And so something that has become increasingly popular as we start to get population scale biobanks where you can cost effectively give somebody like a 23andMe style test and test them for several 100,000 SNPs. And you could get this sort of GWAS appropriate data for them and say, how can we predict your likelihood of having these complex polygenic traits using this panel of things? And you can. And so you take each of these 100B kits, and they all have this tiny contribution, and you add the contributions together, and you get a score. It's like a giant motif. It actually is. It's mathematically adding the contributions of all these things together. How well do you match the profile of having a high heart disease risk? And if you look in the population, you'll get the sort of bell curve distribution of scores where most people have an average phenotype, but some people would be out on the tails and have a better or worse outcome depending on what it is. But you'll have things like I believe this particular study was breast cancer risk, and you can calculate a score of how likely you are to get breast cancer. And if you look at the people who are out on this high tail, they will start to show their risk earlier, and they will have an overall increased risk over time. And something you can do with this, if you're a national health system or if you are an insurance company, is you can say, all right, I have your 23andMe results or the equivalent. I can see you were in this high risk group. It would be appropriate for you to go get screened more often. Maybe you need to start your mammograms at age 40, but in the broader population, that would create too many false positives, and that would cause more harm than good by people, trying to get treatments that they don't need or workups that they don't need. So this can allow you to use these sort of weak hits in order to make a useful prediction in the population. One thing to be aware of in GWAS studies is that you very rarely have purely randomized populations. Things that are adjacent tend to breed with each other more often than not, and so you get this long range correlation because you are not randomizing all of these traits over your entire population. It's not a well stirred mixture, and so you could have a trait that is unevenly distributed geographically or over whatever barrier you have to think about. And because that population is engaging with itself, that you would get local differences in the allele frequencies globally in that sort of concentrated subpopulation that also has the difference in your trait of interest. And so what the symptom of this is you start to get elevated probabilities for everything. Everything starts to look like it's associated because all of your alleles are at a slightly different distribution in your subpopulation because they're mostly breeding with themselves. And it's not just the causative SNP and things near it. So the characteristic sort of signature for this, your skyline starts to look more like Manhattan. It starts to get higher, and this is, I'm going to zoom in on this QC plot right here. This is a valuable way to look at the quality of your of your GWAS results, and this is the same data as in your Manhattan plot, but presented differently. And so this x-axis, this is your this is the expected p-values, and this is your observed p-values, and this is your and you would expect to get your xy. If all your data is well scrambled, you would expect to get your you would expect x to be y. Your observed to be expected. In a good clean GWAS, you would expect most of your observations to be chugging along at the expected line, but only when you have things that are actually relevant, that are actually causal or associated, then you start getting association p-values that are above the randomness line. So these are stronger than you would expect if it was just noise. But if you have the sort of population substructure, you get things where everybody is performing above average. And so there are methods to sort of correct for this population substructure that is above IBO level. And just be aware that it's a thing. Just wanted to show you the sort of this observed versus expected plot. Because this is it's a QC method for looking at your GWAS results. What you really want is things to be puttering along at this randomness line, and then boop, here's your best hits all the way over here, where these are much stronger hits than you would expect to get just by chance. Gene ontology. Okay, so what is gene ontology? What is ontology? It is the study of what entities exist or may be said to exist, and how such entities may be grouped, related within a hierarchy, and subdivided according to similarities and differences. Which that is a lot of fancy words to say that they took PubMed, took all the fancy words in PubMed, and they put this into this diagram thingy. It's a hierarchical diagram that captures PubMed. And so as a hierarchical diagram, you can see here's an example of one. Hexose biosynthetic process, that's a thing that you might want to know about. And that is a subtype of these things, which are, again, a subtype of carbohydrate metabolic process, which is a subtype of metabolic process. And this is structured data, PubMed in structured form. And you have resources that will allow you to browse this hierarchy. Here's a very popular one, geneontology.org. And I can show you what the data looks like. I made these slides back in 2020 when I was working on a microarray for COVID risk, and have the biology Olympiad background. I got dragged into curating the gene list, because at that point in April 2020, literally half the human genome had been postulated in some way or another as a COVID risk. So I got to do the biology BS filter, and aldosterone was one of the candidate pathways. So I was actually looking at this. So what genes are involved in aldosterone? And you have this, and you have these search terms that are things related to aldosterone. You have positive regulation, negative regulation, regulation of blood volume by aldosterone, and each of these come with their corresponding list of genes that you can download. You'll get the other use case the more common use case is you have your list of genes and you want to figure out what they mean. And so I am picking on these guys, but this is very common. What you do is you they knock down their favorite cancer gene. They're looking at things that are differentially expressed in cases where they have knocked down their favorite cancer genes, and they looked for, given this gene list, what words show up in this list. What processes are more likely to show up in that gene list, just over chance, and you'll get these sort of cancer-looking words. Ooh, cell motility, apoptosis, and you say, oh, hey, this really is my favorite gene. Really is the cancer gene. And of course, as always, when you have these large data sets, you kind of have to be careful to not find Rexor, is you can often make them, you can find what you want to believe in them, but at the same time, these large data sets with weak signals in them can also do very powerful things if you use them with humility. And that's the job of the bioinformatician to impose humility where sometimes none exists. Speaking of talking about protein folding, and really it shouldn't work at all, because look, too many degrees of freedom. Look at all that, all these bonds, they rotate. Hey, yes, a Ramachandran plot. But it turns out that that protein folds live in characteristic orientations, where these bond angles take on some stereotypical values. There's this cluster of rotation angles. You get this beta sheet. You get a different cluster of rotational angles in these bonds. This backbone bond. They will settle comfortably into an alpha helix, and this is going to help to reduce the search space to how these fold, because instead of having this infinite array of positions that this bond could be in, it tends to settle in these comfortable positions for it. But even if you are treating these things as, okay, it's got like 3 positions, 3 to the n is a lot. And it's kind of impressive that proteins can work at all. And we do have things like chaperones to try to increase the odds in real life. But if we are doing computational modeling, that we don't necessarily have that luxury. And so we use some strategies to try to reduce the search space for our computers. We can try to look at secondary structures, look at the amino acid frequencies within different types of secondary structures, so you can predict, maybe this alanine wants to be in a helix, and use those to say, okay, I'm going to try to assume that this is a helix or that this is a beta strand. And you can do homology modeling, where I say this sequence looks a lot like this other protein that I have a structure for. I'm going to start with that structure for this domain. And you can take your slightly different sequence and try to thread it onto an existing model to do a of alignment in 3D space. If you really want to, you can try to simulate the atoms, but that's in all but the very smallest cases, that is just not going to happen computationally. And so you have to rely on these hacks and on this, using the existing structure data that you have and trying to exploit known substructures and the known tendencies of particular sequence of amino acids in order to try to get something to converge. Because even if you have your choice of 3 bond angles, or it could be a helix, a strand, or a turn, 3 to the n is a lot. And so these are some practical things that you have to do in order to make this work. Okay, BLAST. What should I have? Okay, I have enough time. All right, so I want to do local alignment of sequences. Let's do it the smart way. Doing it the way I showed you with the Smith-Waterman and the full grid and the exact answers. It's just computationally expensive. In some sort of, what if you have your real world metagenomic database, and you have 2,000 genomes, whole, even bacterial genomes, and you have sequenced your soil or whatever, and you're trying to figure out what's in it. Trying to do that comparison would just take you until the heat death of the universe. And we don't want that. We want answers now. And so we need heuristics. And the one that everyone uses and likes, BLAST, basic local alignment search tool. And the way that BLAST works, here's the intuition. You start with a seed, and you break up your query sequence into words, which are generally, for a protein, that's 3 amino acids. For nucleic acid, you can start with 11. But you are taking this word, an 11-mer, call it, and you are taking that short-mer and you are looking for matches in the database. And it's just a little baby. You can usefully pre-compute, all right, where is this 11-mer in your metagenomic database? Here's your list of seeds. And you want to extend your seeds. All right, so you found your 11-mer, and now you can start extending from that. How well do the flanks match? And you want to extend these things, and you'll get your high scoring segments. And what you're looking for is high scoring segments next to one another. You've thrown all of your seeds at this database. It's good if the front part of your sequence binds to something that is near the back part of your sequence. You can extend these together and get a bigger alignment that scores better. And the results that you'll get out of all of this, you'll get your E-score, your expectation score. What is the expected number of hits of this particular quality given the size of your query sequence and the database? How likely is this to have occurred? All right, and so what types of BLAST are out there, and what might you use them for? There are the obvious ones. You have your nucleotide to nucleotide. You have your protein to protein. You have your sequence, and you want to find others like it. Fine. And so in order to talk about proteins, we got to go back and take a detour into substitution matrices for protein alignments. We talked about scoring functions for DNA. That's fairly straightforward. Proteins are more complicated because there are all these amino acids, and some of them are like one another, and some of them are definitely not. And so you can fairly comfortably substitute amino acids with the same biochemical properties. You have a little hydrophobic, it's a leucine, it's nice leucine, okay. But if you have an acid and you're replacing it with base, then you're probably going to mess things up. And so you have this table of how likely a particular substitution is in related proteins. And so this was compiled from data, and this is being used as a scoring function. And so this is in recognition of the idea that not all of these amino acid substitutions are equal. And there are two common ones that show up. There's PAM, which is based on doing global alignment of very similar proteins. These are things that are over 99% identity. And you're looking for mutations in the closely related. And then in order to get a higher PAM score, because most of the time you're looking for more than 1%, then you just extrapolate it from those closely related ones, just multiply it by that much. And then the BLOSSOM, it's taking the other approach. It's based on local alignment of very divergent proteins. So the BLOSSOM 62, which is kind of the common one that is used for divergent proteins, that's under 62% identity for the input set. And so that one makes a little bit more sense for you're starting with the divergent. You might want to use a PAM matrix if you're looking for something in the 10-20% range. But these are just different approaches for the same sort of figuring out what is a conservative and what's a radical substitution. Okay, so we have the obvious BLAST. You can also do this position-specific iterated BLAST. And that is something that you would use to find distant relatives of a protein. It is building a motif from multiple alignments with a close relative. And it's trying to find that motif to figure out what's important, what is being conserved in this family of proteins. And you can also get this nucleotide 6' translation case, which is useful for very distant relationships. And so you can take your nucleotides, translate it into proteins in all the available frames. It's exactly what it sounds like. And then you can compare it to either the protein database, look for proteins that would be in any of those frames, or you can take those back into nucleotides in the 6 frames and see if there's any similarity at all at that point. Of course, this is a much bigger computational test. It takes longer, but it's useful for very distant relationships where you expect really low conservation. Maybe you can identify it that way, where there's conservation in the protein sequence, even if the underlying codons are changing. All right, for the last, good, we have 15 minutes. This is perfect. You have a practice BLAST exercise. So you want to fire that up and take a try at that. You can try it to BLAST in your UCSC genome browser. And I will be around if you have any questions, we can talk about it.
Sorry, how would I find the start and end positions once I've put it into the BLAST program?
And so you would look for, you're looking for the alignment to HG38.
Yeah, I found the hit on chromosome 9, but I just don't quite know how to read this diagram.
All right, so if you look at this, here, let me show you. All right, over here. All right, so this is part of your BLAST results, and you're looking at, this is this NC blah, 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 chromosome 38 primary assembly. That is chromosome 7. That's all of it. And then so your query sequence, you're going from 1 to 240, but your chromosome, you're going from, here's your start position, here's your end position.
Oh, okay, I see.
Yeah.
Thank you.
And if you look, this part also has a contig sequence. This happens if there's variability. Sometimes there would be amplifications or genomically unstable region. You'll get contig sequences that have an alternate form to them. This isn't the official chromosome 7, but it's a region that is it's a variant that is known, and so you'll have the sequence also shows up in there. It has different coordinates because this is it's not all of chromosome 7, it's just a chunk that varies in individuals. That's what that is down here. But if you're looking for the whole thing, you want this G38 primary.
What do the query numbers mean again?
Sorry, say that again.
What do the query numbers mean again?
That is what position it is within the sequence that you gave BLAST. You gave BLAST this 240 base pair sequence.
I see.
In this case, it's a perfect match from beginning to end, but maybe this front part didn't match to it, and so then your query would start at something in the middle. Like, it doesn't have to match the entire query.
Thank you, everybody.